You know, I'm not sure how many folks kind of join a lot of societies like this for the large macrofungi, but you know, for this talk, we're going to be kind of zooming in on some of the tiny stuff. But I think a lot of that tiny stuff ends up being pretty big in the long run. And yeah, so the work that we're going to be talking about today is actually one of the published first chapters of my dissertation. Pat mentioned, you know, this is all done at Oregon State, you know, working with Joey Spatafor and Posey Busby. But I really want to highlight the input from Dan Thomas and Biddy Roy here, our co-authors. Without them, this project literally would not have been able to actually happen. And I also want to note that I want to shout out to some of the previous you know, foliar mycologists, specifically George Carroll, Martha Sherwood Pike, definitely Jeff Stone. His work has been crucial, you know, for me to even just understand the system, and Mary Bernstein. So yeah, I would highly recommend reading their old stuff, or all their stuff. Sorry. Yeah, and if you're a little confused by the title slide, I think that's good. Hopefully things will make a little more sense later. And so, right, so let's look at coastal Douglas-fir, or Pseudotsuga menziesii, variety menziesii. Coastal Douglas-fir, I don't have to really explain it that much to y'all, but it's a dominant of the Western North American forests. And its range is shown in green on the left here, with the bluish green being kind of the Glauca variety. So everything we're talking about is focused on the coastal variety here. And so it's a mycologically rich host. It's got over or close to 600 different pathogens, and then a less well-documented number of ectomycorrhizal associates and other symbionts that might not really cause a lot, you know, diseases. But even just going off the diseases, we've got a lot to work with here. And obviously in the North American Pacific Northwest, it's found growing in a plantation setting, but these naturally young, mature, and old growth stands can be found, you know, throughout its. And so, like, as these stands of Douglas-fir age, these tree crowns kind of break up and disarticulate and fill back in again. So you'll have a leader branch kind of get disrupted, and then a second leader will pop up, and then you'll have other lateral branches reiterating and filling in space. And over time, you know, this can actually lead to pretty divergently structured canopies. And so, you know, with this canopy on the left, we can see is, you know, growing in a much more, or this crown on the left is growing in a more exposed context, whereas this, you know, this focal tree on the right here is growing in a much less exposed context, with less exposure to the sky above. And so, you know, there's these site level differences, but we also have the within tree patterns of exposure. And so obviously at the base, where there's less exposure, we generally see increasing leaf wetness, where the foliage is wetter for longer periods of time, and it's obviously exposed to less direct sunlight. And then as we move up to more exposed portions of the crown, we see increasing leaf and air temperatures, as well as obviously more intense light. And so further, with Douglas-fir being an evergreen conifer, it's going to retain a portion of the needles that it's made in previous years. And so this age class 4, you know, that is being shown in this drawing here, this is going to be, you know, some of the oldest foliage that we've got on this branch here. And then as we move towards the tip, we get younger and younger, more recently produced foliage. And so there's sort of a temporal exposure gradient that we've got, as well as this physical, spatial exposure gradient that we have on top of this. And so, really, it's possible that all of these factors that I've mentioned so far, can interact to affect the communities of fungi that actually live on those needle surfaces. And so I've got kind of a smattering of them here, but probably some of the most reliably associated. And so first here, we have this Zasmidium pseudotsugi, which is something I've been able to photograph recently, which is actually going to live mostly superficially on the needle surface like this. But everything else that you're seeing here, as you might see from some of the micrographs, these fungi are often trying to actually get into the needle, where it might be thought of as like a more protected space, or at least a different type of dynamic in which fungal interactions can occur. And so, these fungi are called endophytes if they actually remain non-pathogenic for multiple seasons. It's pretty squishy definition, honestly, but the ones that probably fit the definition best are actually going to be some of these Philistictus species, as well as this species, Rhabdocline parkeri. So probably one of the most common right there, one of the most common foliar endophytes. And then we kind of have this, more on the edge case where some of these fungi, such as this Rhabdocline pseudotsugi, where you can kind of see some of these, the ruptures in the epidermis of the needle, and where you can see these plugged stomates in these needles. Both of these foliar fungi can actually cause disease under the right conditions. But for the most part, and historically, this fungus, this Nothophaea cryptopus, I'm probably just going to call it Noga or Notho or Nothophaea cryptopus. It's a huge name. This has not historically caused disease, but it has only, I'd say, in the last 30 years or so, started causing disease, even though it's a native fungus. So given all these dynamics and the potential for interaction, we were obviously interested in determining whether communities from more exposed or open crowns differed from those that were less exposed, or what we'll call closed crowns or canopies. And so, as described in our article published in Fungal Ecology a couple of years ago, we were able to shed some light on this question with an excellent hard-earned data sample set from Dan Thomas, so my co-author on the study. And so really he and his crew and other folks associated with the Andrews, the Asia Andrews experimental forest, were able to climb and sample 7 old-growth trees. So most of these tall ones here, and then 1 plantation age tree, which is this little one that I've colored green here. And as part of that collection, they collected at regular height intervals within these tree crowns. And at each height, they also sampled those 4 different ages of needles that we saw earlier. And so we have that for every tree for the heights that, that are sampled. Right, so here's just like a little snippet into what the life of a single rope climbing tree climber is like. So imagine this repeated, 7 more times. Very difficult ascent process. There they go. Wow. And they haven't gotten to the canopy yet. So yeah, so there's kind of a little bit of that. And so after Dan had collected these samples and kind of reached out to us to, can you actually do something with these so that we can really just start figuring things out? Oops, sorry. He reached out to us about working with his samples, and this is where my fun began. And so here I'm going to kind of introduce this term of metabar coding, or you know, if we're gonna go with the theme of scanning for life forms. Really this technique relies on the fact that we have a lot of these DNA barcodes for fungi, um, and that we're able to amplify and sequence like the entire barcode region or portions of that barcode region in order to identify those fungal taxa. Um, and then we can compare our sequences to those databases and databases of fungal sequences that exist so that we can more or less figure out who we're actually working with. And so our technique relies on doing this in a high throughput manner, which is what metabar coding is defined as. And so that begins where we surface sterilize and get rid of all the surface contaminants, the DNA contaminants on the surface of the needles, so that we don't get anything that's just kind of randomly associated or spuriously associated with the needle. We freeze dry them, and then so they break up nicely, and then we'll extract the DNA from them. We'll amplify this ITS2 subregion, which is, if you guys are familiar with, do a lot of barcoding in this society, then you'll realize that this is only a portion of the full barcode, but for this aluminum MiSeq sequencing technology, the trade-off is that you get nearly 25 million sequences instead of maybe just your few barcodes with traditional Sanger sequencing. And so we use all those millions of sequences to kind of survey the foliar community, mainly because a lot of these fungi will not culture for us, and a lot of them might only be detectable via microscopy. And even then, you might just be looking at this hyphae. This hypha looks very similar to this other hypha under the microscope. I can't make an ID. And so this is a high throughput way to actually perform this identification. And so we'll compile these things that we call operational taxonomic units, which are our best guess at the unique taxonomic entities that occur in our samples. And this can kind of range from somewhere to a genus level to a subspecies type guess. So it's pretty squishy, but it's the best the field can really do. And then once we have all those OTUs, we can look at the relative proportions of each of those in every sample to characterize our communities. And so we still need to kind of come back to and define this idea of exposure, which I've kind of used in a very loose sense, but I think you can maybe see why. So for us, we figured we could break this up more easily into 3 different components. So height, age, and closure. And so, so here are 2 trees that are shown at the same scale. And height is pretty intuitive. This is just that recorded height at which the samples were taken in each of these tree crowns. And we already had that piece of information when Dan had collected these needles. And then additionally, like I mentioned, we'll have our needle age component. So these older needles will have been more exposed to whatever environment they've been exposed to, more so than these young needles. So these young needles are less exposed to the environment. And then, defining canopy or crown closure can be a little bit more difficult or tricky, especially if we didn't measure it in the field in our case. So you can use more traditional techniques like this hemispherical photography based approach, or you can use tools like densitometers to measure how much light is kind of reaching a given field of view that you're measuring from. However, if you have the geographic positions of each of those trees on the landscape and forgot to or didn't consider taking those measurements while you're precariously climbing, up in the tree crown, you can rely on light detection and ranging, or LiDAR, to kind of get you a similar, but it's a little bit of an inverse measurement where you're actually kind of measuring the amount of intervening vegetation and less so the amount of light reaching a point. So Probably not as good as traditional techniques, but in terms of the amount of landscape it can cover, as you'll see, I think it's an improvement. And so, you know, LiDAR has gotten a, oh, am I going to get hit with a commercial? Hmm. Well, it's not essential, but the idea is that LiDAR has gotten, a lot of press recently in the last few years in the context of things like self-driving cars and most recent iPhones maybe is probably a lot of people's experience. But the technology is actually much older and even more versatile than those implementations seem to suggest. So in fact, like these airborne LiDARs have actually been used since as early as 1979 to profile various forest attributes, as we'll see here in an airborne context. Whoops. There we go. And so briefly, this airborne kind of LiDAR, it's kind of weird pointing a laser at a thing shooting a laser, I guess. But this airborne LiDAR works by shooting these laser pulses from known like geographic positions and at known firing angles so that it can geolocate any one of these returns that it might get as that laser pulse bounces back. And so, here our platform is this Cessna, which is pretty common for a lot of airborne lidars, but you might see drones increasingly used to do this. And so the main idea is that targets that or that returns that take longer to get back to the source of the emitter are interpreted as being farther away in three-dimensional space. And then another factor to kind of appreciate how cool LiDAR is that it's not just a one-for-one, a single pulse gets you a single point return, but this single laser pulse, of which you're shooting millions, can actually give you 1, 2, 3, 4 peaks as that beam intercepts the different parts of the crown here, from this tree, from that tree, and then the bare earth right here. And so, if you keep flying around and shooting enough lasers, you'll eventually have these like millions of data points with which you can actually build a representation of whatever forest you're surveying. And the use is pretty apparent. So from here, a lot of foresters will derive things like biomass, canopy closure, crown closure in the case of stuff like what we're doing, and sometimes even tree species can be determined from the shapes of crowns and the other spectral data that you get sometimes whenever you're using LiDAR. And what I think is really cool too is that it's not just a toy, but like it's even been used to describe the microenvironments for macrofungal communities. And so, as part of this this Danish macrofungal survey, um, employing lidar data, they were able to find that increasing amounts of canopy closure were associated with increasing macrofungal richness. So, more species, more taxa detected in a given site. But not only do you get increasing richness, but you actually just get changes in the overall composition of the community as you move from less canopy closure to more canopy closure. And so, for our purposes, that's nice, but if you can't find the data, what's the point? Turns out, really fortunately, I was just kind of cruising the Digital Coast Data Access Viewer that's operated by NOAA, looking at some of my sites, and sure enough, like for the same year that sampling began, we actually had a full coverage LiDAR data set that we could use to describe the canopies and crowns of all of our trees. So definitely turned out to be one of my more productive pandemic projects for sure. And right, so using the LiDAR package, which I'm kind of shouting out here, I really recommend it. It's free to use, like all of our programming software is. So if you go to NOAA and download that that resource, you can start playing with it using this package. You can't get these visuals though. So right, so the first part of this process was to actually kind of just create these little cylinders around each of the trees here. So more obviously centered on tree, but there is a focal tree in the middle there that we're focusing on. And we get 10 meters radius around that center. And what we'll do is we'll first calculate the number of points above our whatever height we're interested in and getting a closure measure for. And then that will serve as our numerator. And then we'll come in and we'll divide that by the total number of points in the cylinder for each of the cylinders surrounding each tree. And then we'll apply a scaling factor to that ratio. But in the end, this will give us height closure profiles for every tree that we had sampled in the data set here. And so I want to kind of call out a comparison that intuitively made us feel good whenever we got our crown closure indices back. But you can see here for the our smallest plantation age tree in the data set, it's associated with low closure readings across its heights. But our tallest old growth tree in the data set had the greatest crown closure at every height here, and they were the furthest separated from each other. So that lines up with our intuition pretty well moving forward. So okay, we've got our tools, we've got our covariates and things to analyze with. We got to come back to this question though about how whether communities from open crowns or closed crowns are different from each other, and whether these communities correlate with the amount of exposure. And so looking at that OTU richness, so these are kind of those taxonomic guesses, we actually see that increasing amounts of closure measured on the x-axis here were correlated with a higher species richness, or OTU richness, excuse me. And that we also saw, you can kind of see the lines colored by the different needle class ages here. We also saw that the older the needle was, the richer it also likely was, which is a fairly intuitive result too, but it was nice to see how that may be interacted with our defined closure index. And so interestingly, this is our best performing model, and it did not include the height that you were actually, of the sample collection at all. So it really seems you can at least model richness pretty well with just measuring crown or canopy closure and needle age. So overall, I mean, we like more variables, obviously, but for what we had, we don't need height. So overall, we have obtained 218 of these taxonomic guesses, these OTUs, across 256 samples. So, as far as some of these high throughput sequences, these high throughput sequencing studies go, this actually isn't very rich. And when you look at the top 95% of reads for each tree here, so each column represents the total reads associated with each tree, the total number of sequences that we got. We can actually see that these communities are pretty strongly dominated by two genera. So here we're coming back to our rhabdocline that I mentioned earlier, as well as the Douglas-fir cryptopus. And both of these sections are entirely represented by Douglas-fir cryptopus gallmanii and rhabdocline parkeri. And so these make up the vast majority of the Doug-fir foliar needle community. And then when you look at composition a bit more, we find that communities largely group by the tree they came from. And so this is a community analysis technique where each of these points here represents a different sample, and we've colored them just by the tree that they came from. And so what we actually have here, we can see, that the points are, in many cases clustering according to the tree they came from. But that's not really, all that's going on here. Whenever we perform, compositional tests that actually look at the interaction between height and closure, we actually see that, again, height plays no role, apparently, in the structuring of these communities. And we have a fairly low R-squared here. This is some of the more conservative ones that I'm showing you guys, for the whole study. But we're actually able to see that for certain needle ages that this can be as high as 13%, 0.13 coefficient of correlation. So it's very age dependent is what we found. But I think the bigger take home is that, again, height does not really seem to matter. It's what seems to matter more at the in terms of the data that you put into these analyses is, the actual measurement of closure and needle age. So using the entire Andrews forest to bin our samples into basically open samples and closed samples within and across different tree crowns, we found that our Nothofae cryptopus, our Noga, was identified as an indicator species of these open parts of the tree crown. So if you were to randomly sample some needle foliage and you found Nothofae cryptopus, you might be able to reliably say that I'm probably looking at a sample from an open part of the crown. And the opposite is true for those Rhabdocline parkeri OTUs. Oh, I got the color switched from earlier, but I think you guys can follow. But those Rhabdocline parkeri OTUs we found were associated with closed parts of the tree crown on average. And so let's look a little bit more about what that would look like when you look at the relative abundances for both of these species. So if we follow the relative abundance of Nothofae cryptopus, we can kind of see that for this closed group, so kind of just pay attention to this closed group, the left side of these plots that we have now, we can see that we're getting this overall slight increase in the relative abundance of this fungus. But then when we look at the open group, we can see that this increase is just much more pronounced. So all of these, we still kind of get a slow start for H class 1 and 2, but the switch from 2 to 3 is very pronounced. And so, and that's carried over into H class 4. Contrast this with our Rhabdocline parkeri friends that we identified earlier. They like the closed parts of the crown. When you look at open parts, you can kind of see it seems to kind of just peter out, as we go from young needles on the left to old needles on the right here. But when we look at that preferred part of the crown, we can actually see that by this H class 2 for that closed section, we get this huge increase in the relative abundance of Rhabdocline parkeri. Yeah, so the question is kind of like, what's going on here, and why might we kind of see these two different fungi take pretty different paths? And so it's possible that really any of these trait differences might lead to these two different species just occupying totally different niches, and leading to the patterns that we've seen in the sequencing data set. But really, I think we're going to want to focus on this idea of that Rhabdocline parkeri even though it gets into these Douglas-fir needles, it gets in very early, but then it does absolutely nothing for sometimes up to 7 years, and maybe even longer in the case of an extra long-lived needle. It will totally just ride out the life of that needle, and then once that needle has died, it will begin degrading that needle, saprotrophically. And then we'll contrast with that with Nothofaea cryptopus, which is a very well-defined biotroph. So it needs a healthy living host to actually kind of be leaking nutrients into the needle that it can just sort of lap up. And so my guess is that, these closed sections of the crown actually are able to catch more dead needles from higher up in the tree crown, and that those dead needles that contain the rhabdocline parkeri will then serve as inoculum sources at wherever they fall in the tree crown. So potentially living this totally, tree-borne life, within the canopy. And then Nothofaea cryptopus is, prefers those open parts of the crown because that's where the most photosynthesis is happening. That's where foliage is most physiologically active. And so, at least on the plant side. And so, this makes it more optimal in terms of just a resource capture angle for Nothofaea cryptopus. So where do we go from here? We're going to zoom out like a lot, but I thought it might be easier to kind of make like a lame Star Trek comparison. And so I'm sure everyone has at least seen part of a Star Trek episode, or at least a parody. But like practically every episode where they come across a new planet, they're traveling through space, and then the captain says something along the lines of, let's see if that's going to work for us. Scan the planet for life forms. And the way this technology is used in the show basically functions like magic. The writers kind of just make up a sensor that matches what they need to measure, and the crew can kind of have an idea of what they're actually getting into, before they embark on the planet. But but I'm not saying I'm definitely not saying we're at like Star Trek level, technology and understanding of nature just yet. But the diversity and number of satellites and other instruments and sensors that we've now got kind of pointed at Earth is, it's getting to be it's reaching a high point, and ever since we've launched ourselves into space, basically. And so many of the platforms that are actually kind of shown on these on this outer ring here, those are still active in monitoring and acquiring data that we can use to monitor and study Earth's biosphere, but usually at different like resolutions and stuff. And so as LiDAR technology continues to kind of mature, and it becomes a lot more accessible and affordable for researchers to use, I think we might reach a point where we can scan a planet, probably ours, and infer what kinds of plants and fungi and animals even might actually be at any given location, based on the types of remotely sensed data that we get back. And so obviously making those connections between this remotely sensed data from satellites and planes and other types of scanners, like to make use of that in like an ecologically relevant sense, we need to be just linking more data sets to these variables that we're producing tons and tons of every year. I think it's a long way to go before we can understand how different organisms prefer different microenvironments and different microclimates. But I'm hopeful that we can get to this point of application. And I actually think that the Douglas-fir foliar system, fungal foliar system, is a pretty good use case of this. where you're combining remote sensing technology, potentially LiDAR, and fungal ecology. So I'm not sure if y'all are aware, but in a lot of like low-lying areas of the Coast Range and the foothills of the Cascades, we actually have a lot of Douglas-fir stands that are affected by a foliar fungal disease called Swiss needlecast. And this Swiss needlecast is caused by that Nothofaeocryptopus, we've been talking about throughout this talk. And, since 1996, we've had aerial surveys increase the size, the acreage of affected Doug-fir, or the area of Doug-fir that's actually affected by this Swiss needlecast, this S and C here. And really, that seems to be in response to increasingly warm winters, which Nothofaeocryptopus really responds to. And it's probably the single climate attribute that that you could model most of Nothofaeocryptopus's spread by. And so perhaps by combining these aerial surveys, which are great, amazing data sets, but incredibly expensive to acquire, and require a lot of trained personnel to be able to identify a disease stand from a really fast moving plane. Perhaps we could combine these surveys with LiDAR surveys as well as satellite LiDARs and other sensors to better model the spread of this disease on the landscape. So it's likely going to just keep increasing and reaching higher and higher elevations, and more Doug-fir will be, over long periods of time, choked to existing at high elevations on the landscape. So yeah, so just some broad conclusions here. I think I've probably harped on this enough, but LiDAR and endophytes have the potential to bring us some new insight we didn't have before. Not just at these small scales, but where the trees were actually sampled, for instance, but as far as that LiDAR survey exists, you might be able to extrapolate and estimate the distributions of fungal communities. So I think that's a big goal for me on the horizon in the field. And additionally, it seems like at least in the Doug-fir system, closure and needle age seem to matter most, and the spatial component of height is sort of seems accessory to the actual measured microenvironment that you get with something like canopy or crown closure. And definitely a Doug-fir specific result. These are Doug-fir specific fungi, but they dominate all our entire sample set. And what we've seen, based from this sequencing data, is that they actually seem to inhabit different niches. And yeah, and the last point I think is just to make a call to fungal ecologists to, I'm glad I've observed this pattern, and we wouldn't know this without this study, but I think we also need to start doing more ecological experiments to test some of these mechanisms, like the one I gave today to explain these patterns. That needs to be tested. And so with that, I would like to acknowledge my funding sources. This is going to be kind of a fake out ending, but down below was what funded the immediate work that I showed you guys. And I want to thank all the lab members in the Spatafor and Busby labs. I also have one of the most awkward clips from Star Trek that inspired the title of this talk, so we'll see if this is loud enough. Whoops, I click it. Gotta do the loop around. Oh man, I can't find my mouse. Oh, there it is. Okay. Energize. That's what I say a lot, yeah. Would you believe that Brent Spiner improvised that? But so where does OMS actually come in? No. So actually, what we talked about is just that first chapter, but I'm going to give you guys like a quick sneak peek about how you guys have actually helped me generate data for my my two last chapters, and they're probably my two most demanding as well. So our last sample set came in around 256 samples. The sample sets associated with these two chapters, add up to around 4,800 different needle extractions or needle samples that those lead to extractions, those lead to PCRs, et cetera. So we're in the high thousands here whenever you add everything up. And we have a lot more individual trees that get sampled. Some get sampled, over 3 years. But then we also expanded the number of sites in the PNW. So we're not just talking about the Andrews, we're talking about Southern Oregon up to Southern Mid Washington. And so, really, by the time that we're all done with this, I think this is actually going to be one of the largest studies of its kind in Douglas-fir, and probably several other, it's probably beating out several other plant hosts. Not that I'm, concerned about that, but but I think it'll be fun to kind of work with a data set this large. Here's just a sneak peek of the results that I have from a test run using many fewer samples, but it's using the technique that the funds you guys gave me, helped me develop essentially. And so the main thing that we are actually getting with this new technique is an estimate of the fungal abundance, not just the relative abundances that we've been talking about all this time, but some kind of idea of the total fungal load that a needle is experiencing. And so each column here that's going along the x-axis and in this figure, that's associated with a sample, and they're grouped into this coastal garden samples and this montane, Douglas-fir garden as well. And when we're looking at the bottom, we have the, just the relative abundances that are associated with every sample, like I was showing you guys before. That was at the tree level. This is at the individual sample level. We can see pretty strong compositional differences, but this is not what the new technique gives you. That's what the standard technique and way of doing things gives you. But with this abundance technique that I've been able to implement with y'all's help, we can see that the differences between that montane garden and that coastal garden are actually even more pronounced. And that even just looking at the compositions here, we probably underestimated the differences between these two sites that are on the same latitude, but have a latitudinal, coastal, continental sort of gradient that they're following. And so I'm really excited to see what the full data set looks like. That was dropped off at the sequencer last Tuesday. And so now I'm just anxiously waiting for 500 million sequences with this technique. So yeah, so I'm also open to actually giving another talk too once I have those data as well, because those will be new and fun to play with and share with folks. So yeah, so that's it. I'm down to take questions too, and we can, yep. And I can go to any slide anybody wants to, so. No, so there's actually a master's student, Leela Gonan, who she's already moved on to, from OSU, but her thesis was focused on we actually sampled the same, sorry, for for all of this work that I'm mentioning with these thousands of samples, she sampled in that same network of Douglas-fir trees. And her sampling method actually involved shaking the trees and dislodging a lot of those needles. And she does find Nothofeia cryptopus, the supposedly biotrophic fungus, still in these needles. Whether those needles were just on the edge of, barely attached to the tree versus already fallen off and, existing for a long time, I don't know. I don't think her method can really resolve that. But you have to imagine that it's probably like a normal distribution of, some are old needles, some are young needles, but most have probably been hanging around for a little bit in the canopy. So, and she does see Rhabdochyme parkeri, so that was good. That's what I really expected to see, was more the saprotrophic fungus in her data set.
So were those age classes you saw, or were those 1, 2, 3, and 4-year-old needles?
So, right.
And your microphone is no longer working.
It's not? Has it been a problem, or have? Okay, good. Let's try that. Can you hear me? Okay. So, oops. Hello. Oh, yeah. That's what we've been missing. Sorry, could you repeat your question again?
Was it age classes? You had A1, 2, 3, 4, or are you looking at specific age?
Right.
1-year-old, 2-year-old, 3-year-old.
So these are definitely age classes. So what the main inference there is that we're not talking about like full year complements of these needles, from spring to spring. So in my case, what I was calling A1 is actually a spring to winter age class. And so it hasn't made like a full cycle. It's in its first year, it has not completed its first year. Yeah, so that's the way to, I would.
So nothing was older than 4 years.
Well, I mean, based on that, it's going to be more like a 3.5-ish type thing. Yeah. So, and then our age 1 is probably coming in a little closer to, 6 to 7. Yeah. Good question. Yeah. Yeah, that's yeah, I can repeat it. So the question was, what's sort of the tipping point for the fungus to be switch from being somewhat benign to being pathogenic? And so I guess there's kind of like the first big main switch. So the first people to start seeing that Swiss needlecast was a problem were probably Christmas tree growers in the 70s, late 70s. And, really what they like, up to that point, everyone, or, the mycologists knew that Nothofeia cryptopus was everywhere, but it just didn't cause disease. And so really, the only thing that that people have really been able to reliably connect to that switch has been the increases in warm winter temperatures. And so we've, at some point in the 70s and probably a little earlier, we crossed some threshold of optimality, for the Nothofeia cryptopus, where the warmer the winters are, the more that it's actually like what Nothofeia cryptopus is trying to do in winter. It's trying to make structures to produce spores for the spring. And so the warmer your winters are, it kind of kinetically is just going to speed up the growth of the fungus in the needle. And so by that following spring, if you got a lot of nice warm winters or winter temperatures, you're probably making a lot of spores. So yeah, and then I think it also hasn't helped. A lot of what's been pointed at too is the increased planting of Doug-Fir in what has probably historically been like a coastal spruce zone. And so what's kind of fun, I mean, it's not funny, but because of the scale at which it's happening, that a lot of foresters are actually having to look at planting more spruce on some like just really non-optimal sites, for Swiss needlecast. And it's, in some cases, it might just be like, we're going back to the spruce zone, after this sort of unnatural, continued growth of Douglas-fir in areas where it just hadn't been grown. So, right, yeah.
Kind of old school, but I think of MacArthur's work with birds where
Islands.
pushing layering. And then I think of Samard, which might be a little bit more recent. She talks about the importance of diversity in the stand, of not just old growth Douglas fir. Talk about those two types of things. Whether you think about that.
Yeah, so sort of having these layers of diversity, and then also, could you repeat that last part with Samar?
Samard, British Columbia,
For ectomycorrhizal, yeah.
Finding the mother tree.
Yep.
Talks about productivity in the forest stands, and diverse stands with deciduous trees and greater than just monotypic stands of Douglas fir, old growth.
Yeah, so what's actually pretty helpful, I feel like I'm going to take the last part first. Right. So a lot of times with a lot of some of the grassland work, some done by David Tillman in the Cedar Creek Preserve, one of the big focuses there is sort of this diversity productivity relationship, where where increasing amounts of diversity are associated with greater productivity. And here, I think in like the context of a plant associated system, I think you could think of it in two different ways. Where like what we're seeing a lot here is that, granted, this is a subset of the data, but we're seeing a little bit more fungal load. So if you look at the left here of the coastal garden, we're seeing higher fungal loads associated with less diversity. But this might be just a different, outcome. Like we might not expect a diversity productivity relationship as what Tillman or Samard may have described, because of the way in which, we might just be getting higher dominance of these pathogenic species. Wherever they're by themselves, they can just do what they need to do in terms of putting on fungal biomass in the community. Were you more interested on the plant side, though, of that diversity relationship?
Things went through my mind.
Yeah.
I wonder if
Yeah. I'd say like that was one of the main inspirations for developing this fungal load technique, is that we can actually start looking at patterns that other ecologists have been looking at forever. But because our sequencing techniques kind of limit us to those relative abundances for the most part, I think it's actually severely limited the types of ecological connections we can make to to past macroecological works. Working with macroorganisms. So I sort of see like the technique that you guys have helped me develop. I see that as a way to actually bring some of the microbiology into macro terms for us to work with. So that was probably too sprawling, but oh yeah.
I was curious in areas where you saw a lot of
Mm-hmm.
were there any like trees that you would have expected to have that and didn't? And then it seemed like there was any particular like competing species that inhibiting the spread.
Yeah, so I actually think the data set that's going to be coming out is going to be better able to address that. It's technically an experiment, whereas what I described is observational kind of correlative type stuff. Yeah, let's go back to that stacked bar. Yeah, so part of the problem here actually is kind of, I mean, it's not a very interesting answer, but some of the issues in figuring out who antagonizes who comes from the limitations of those sequencing techniques that I mentioned. So one of the problems with relative abundances that have to add up to one is that when one part, when the actual biomass of one component increases, in relative abundance term, everything else has to decrease. And so it's like you're having to, whatever statistical method you use is going to have to account for all this constant, shifting of reference points. And so I think we can still correct for that in this data set. We didn't look at it for this study. But I mean, the only main antagonism that I'm aware of is that this rhabdocline parkeri, which was very dominant in this data set, you contrast that with the rhabdocline pseudotsugi that I mentioned, and I found that in one sample. And so that's actually what I would consider a more lethal pathogen than the notho, because the Nothofacryptis kind of seems to be on this 3-year death cycle, where the needles will fall off by year 3. With rhabdocline pseudotsugi, you'll probably only have 1 year. And so previous work has actually shown that rhabdocline wherever there's a lot of this saprotrophic rhabdocline parkeri, you don't get a lot of the pathogenic rhabdocline pseudotsugi. And I think what we see here, I mean, I detected very little of it for the heights that we sampled. And so, I think that's a potential interaction worth investigating, with like an inoculation experiments where you put one on the plant first and then do the other and then reverse it and then controls. Yeah, so I'd be very interested in knowing if that's a real antagonism or not. But unfortunately, when it's absent from your data set, you can't actually look at some of those correlations. So. Oh, I see one there. I'd say it's generally the ones that grow into the needle that cause more issues for, for the Douglas-fir foliage. The ones for a lot of the fungi growing superficially, like that Zasmidium, which is practically never going into the needle. If it is, it's kind of getting close to those stomates, but no one's described it as just going in and coming out, Yes, yeah. It's actually it's doing a little bit more too. I don't know if you saw in kind of my little indicator here. I have this little bit. I say that it grows intercellularly once it's actually in the needle. But what it will also do is once it's made it in, for some reason that we don't really understand yet, it actually goes back out of stomates and the pores on the needle, and then grows on the surface, and then comes back in. And we'll do that every year, kind of like a sewing machine, kind of just, going in and out. I think the best guess at the moment is that it might help avoid the host's immune system by being literally outside of the needle for certain periods. But no one's I think, has even touched on that.